KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
Neet, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Students, now in this video we are discussing development of Anthar and Microsporogenesis. Okay, so first of all let us see the development of Anthar. So how this particular Anthar development process takes place. After completion of development of Anthar, so we will observe or we will study about the formation of microspores by the process of Microsporogenesis. Now let us let us see one by one. Now first point, a group of cells which are located just below the epidermis. The group of cells which are located just below the epidermis in a vertical rows. In a vertical rows of hypodermal region at 4 corners become large. Okay, means here, so we have started to discuss about the development of Anthar now. So there, first of all, in this particular situation, we will concentrate on the development of Anthar. So how this particular Anthar formation taking place means, so few specialized cells which are present below the epidermis. Below the epidermis means such particular region we can be taken as hypodermal region. So and these cells also how they are arranged like vertical rows. The cells are there in vertical rows in a hypodermal region at 4 corners become large. So here this kind of a point, same point already we have seen in previous video. Next one, these cells are called archisporial cells. Means what are the cells are become very large just beneath of epidermis. Now all these specialized cells we will be calling as archisporial cells. Archisporial cells. Now these archisporial cells divide periclinally to form primary parietal cells and primary sporogenous cells. Means from the archisporial cells, 2 cells of formation we are observing. So what are those? 2 different types of cells names are. So first one is primary parietal cells and second one is primary sporogenous cells. So by which kind of division? Periclinal division. Periclinal division means suppose if one cell is there means parallel cut. Not vertical, right? Parallel cut. Suppose one cell parallel cut only, our subject point of view we will be calling as periclinal division. So there one particular archisporial cell divides periclinally and form how many number of cells? 2. Names are primary parietal cells and primary sporogenous cells. Now let us watch. So what is activity of primary parietal cells and what is activity of primary sporogenous cells? Let us watch one by one. Now primary parietal cells, let us concentrate. Primary parietal cells undergo further divisions. Means what are those? One is a periclinal division and one more is anticlinal division. So primary parietal cells undergo periclinal division and one more is anticlinal division. So definitions already I have given in previous video. So divisions to form a series of 3 to 5 layers making walls of the anthers. So definitely here, so because of the activity of primary parietal cells, so when these primary parietal cells undergo periclinal division and anticlinal division, Periclinal division as well as anticlinal division, automatically so how many layers of cells formation takes place? So 3 to 5 layers of like a series of cells formation takes place. All these cells, so which are involved in the formation of endothelium, and which are involved in the formation of tapetum layers, and involves in the development of tapetum cells. Means here if you see the structure of anther or the structure of microsporangium, so we have seen there are different types of layers: epidermis, endothelium, next one middle layers, and tapetum we see now here already. So the epidermis is there. Just below the epidermis, all these processes are taking place. So means first of all, primary parietal cells undergo periclinal division and anticlinal division. So thereby here 3 to 5 layers of cells formation taking place in anther. No, all these are all involved in the development of endothelium, middle layers, and tapetum. So thereby here first layer means epidermis already is there. Next layers are endothelium, middle layers, tapetum formation taking place from the primary parietal cells. Next one, second like cells, which one? Primary sporogenous cells. Primary sporogenous cells divides by mitotically. Listen carefully. Primary parietal cells under the division is known as periclinal and anticlinal, whereas primary sporogenous cells divides by mitotic division, mitotically divides. So first of all, archisporial cell is there. That archisporial cell divide periclinally. Two cells of formation taking place: primary parietal cells and primary sporogenous cells. Primary parietal cells divides periclinally and anticlinally. So 3 to 5 layered cells of formation taking place in the anther. All are involved in the development of endothelium, middle layers, and tapetum. Later, next cell, that is primary sporogenous cells. Primary sporogenous cells divides by mitotic division to form sporogenous cells. So to form sporogenous cells or sporogenous tissue. So thereby sporogenous tissue is a development taking place from the primary sporogenous cells. When these primary sporogenous cells divides by mitotic division, so definitely leads to the formation of sporogenous cells or sporogenous tissue. So in our textbook, so this primary is or sporogenous tissue only we will be calling, or we will be taking as a homogeneous. So homogeneous tissue, homogeneous sporogenous tissue means, so what are the cells are present in this sporogenous tissue will not show any kind of differentiation. Means here the all cells are seems to be same. Right. Now that point, suppose if you can remember means okay, no problem. Otherwise if you are feeling burden, you can leave it. Now what is the main important bit? Primary sporogenous tissue or the cells are there, divides by mitotically. At the time which one formation takes place? Sporogenous cells formation takes place or sporogenous tissue formation takes place. Later sporogenous tissue differentiate into microspore mother cells. Sporogenous tissue is there, homogeneous sporogenous tissue. So later that will be transformed or differentiated into how? Differentiated into pollen mother cell or microspore mother cell. So already PMC or MMC we see microspore mother cell formation, microspore mother cells formation takes place. Now each microspore mother cell, mother cell means always ploidy is diploid. Ploidy definitely kandipa diploid state means here the definitely 2 sets of chromosomes we can observe kandipa. Okay, so thereby here each microspore mother cell or cell of a sporogenous tissue divides to form 4 haploid microspores or pollen grains by meiotic division or reductional division. This process is known as microsporogenesis. Now here once the formation of microspore mother cell is completed, now what is happening? So this particular microspore mother cell, now it is converted into or the cell is nothing but spore mother cell or sporogenous tissue. That particular microspore mother cell undergo meiotic division. Important, already this particular point we have seen in our classes. but let us see by one more time. When microspore mother cell or pollen mother cell undergo meiotic division, sir, why sir here this particular cells undergo meiotic division means, so for the formation of 4 haploid microspores, definitely there 4 haploid microspore formation takes place, definitely it must be required. That's why these 4 for the formation of 4 haploid microspores or pollen grains. So definitely microspore mother cell, depending on the which kind of division, meiotic division or reductional division. So once the particular when this particular microspore mother cell undergo the meiotic division or a reductional division, so definitely leads to the formation of 4 haploid microspores, 4 haploid microspores or pollen grains. So once if we are observing the formation of pollen grains or the microspores means, such particular phenomena or the process we will be taking as microsporogenesis. Genesis means the development or the formation, likewise we see no. So genesis of which one? Microspores. Microspores are nothing but pollen grains. These pollen grains are developed from the microspore mother cell by the process known as meiotic division or reductional division. Right. Next point. At the initial stage, all 4 microspores means here all means how many number of microspores of formation taking place? 4. No, all these 4 microspores are attached together with the help of callose layer. If one tetrad formation taking place, likewise we see no. If 4 pollen grains are commonly covered by or commonly surrounded by one particular layer, that particular layer name is callose layer. This is important. Means here our knowledge point of view, 4 haploid microspores or 4 haploid pollen grains of formation taking place. In initial stages, all these 4 microspores are attached together with the help of callose layer. Ellame 4 pollen grains. So 4 pollen grains are commonly surrounded by callose layer. Callose layer. In this group of microspores or this kind of group of microspores we will be calling as tetrad. So means in this tetrad, no, how many number of pollen grains are there? 4 are there. Because of the presence of 4 pollen grains as a group, that particular group, our subject point of view, we will be calling as tetrad. This particular tetrad is surrounded by callose layer. Important, listen carefully. Callose layer. Next one, after some time. After some time, what was happened? After some time, this callose layer is dissolved by callase enzyme. This word ends with AS, means stands for enzyme, callase. So that particular callase enzyme degenerates or dissolve the callose layer. Callase enzyme degenerates or dissolves the callose layer. So thereby automatically what will be happening? One more important point. See that this particular callase enzyme which is secreted by tapetum. Actually here our knowledge point of view, tapetum means nourishment tissue which supplies of food materials to the developing embryo or pollen grains. Likewise one important point we discussed and in previous video, and one more important point, middle layers made up of parenchymatous cells, and these middle layers stores the food materials. Now tapetum gets the or receives the food materials from the so from where? Middle layers. At the time only that particular tapetum acts as nourishment tissue or nutritive tissue. No. One more point we are discussing. What is that? For the degradation of callose layer, so which helps in the keeping of all pollen grains together as a tetrad. For the degeneration of this callose layer, callase enzyme which is secreted by the tapetum. Means here for the degradation of callose layer also is under the control of tapetum cells. So that particular point also you try to save in your mind. Now during this period, during this period means from the starting from the archisporial cells to till the development of till the generation of pollen grains. So while this process is going on, at the time what is going on? No, during this period spherical bodies are formed inside the tapetum cells. So here the spherical bodies. So spherical bodies means already we have seen small particles like grains formation taking place within the tapetum cells. In previous video we see now. So those spherical bodies are formed inside the tapetum cells before their disintegration. Before their disintegration. These spherical bodies are known as Ubisco bodies. So here in previous video we see tapetum cells are there within the tapetum cells. So before degeneration of these tapetum cells within the tapetal cells, Ubisco bodies or pro-Ubisco bodies of formation takes place. When these Ubisco bodies are covered by sporopollenin, at the time such kind of pro-Ubisco bodies we will be calling as Ubisco bodies. We see now. So these spherical bodies which are present in the tapetum cells we will be calling as Ubisco bodies. These Ubisco bodies are mainly made up of a complex substance is nothing but sporopollenin. Already yesterday's information only, but newly we are learning now again. So thereby Ubisco bodies are mainly made up of a complex substance, newly made up of a complex substance known as sporopollenin. Now the importance of this particular sporopollenin is, what is the main important point now? The sporopollenin is a polymer of carotenoids. Now here one more point once you see there, this particular one, what is the thing? This particular sporopollenin is a polymer of carotenoids. Once you see that first line means sporopollenin is a polymer of carotenoids. Now after the formation of Ubisco bodies, where in a tapetum cells, after the formation of Ubisco bodies in tapetum cells, what is happening? No, the tapetal layer degenerates. Once you see there, first of all in tapetum cells, pro-Ubisco bodies of formation takes place. These Ubisco bodies by covering of like sporopollenin, which one formation we are observing? Like Ubisco formation we are observing. These Ubisco bodies are completely covered by means of sporopollenin. Sporopollenin is a polymer of carotenoids, is made up of the polymer of carotenoids. Next one, after the formation of Ubisco body in the tapetum layer, so when Ubisco bodies development we will be observing in tapetum cells. So later these tapetum layers or these tapetum cells are degenerated. Next one, Ubisco bodies participate in the formation of the exine layer. If pollen grains are there, these pollen grains outermost layer is made up of sporopollenin. So that particular the outermost layer is exine. That particular exine is made up of sporopollenin, isn't it? So now here what is happening? For the formation of this exine or the outermost layer of pollen grains formation point of view, so there is a involvement of Ubisco bodies. Means Ubisco bodies participate in the formation of exine of the microspores inside the pollen sacs. Means like tapetum is innermost layer out of 4 layers of anther. So that particular tapetum layers helps, or first of all in the tapetum cells Ubisco bodies formation takes place. Now these Ubisco bodies helps in the development of exine layer. So that's why only exine is made up of sporopollenin. Because of the presence of sporopollenin exine, so we will study these pollen grains as of fossils. Likewise some points we learn now. So those points means here even exine formation point of view also definitely there is a involvement of Ubisco bodies participation. So next Now thick walled microspores are known as pollen grains. So after somewhat time, no, definitely these pollen grains means tetrad pollen grains are there, no. All these tetrad pollen grains will be ruptured. Why? Because of the activity of callase enzyme. Callase enzyme, all pollen grains will be splitted each other. So now here what is the point? Now the thick walled microspores we will be calling as pollen grains. Now here somewhat rough diagrams I have drawn. So let me explain. So just 1 or 2 minutes. So how this particular development of Anther, and here this particular diagrams are the development of Microsporogenesis. Now let us watch one more time. Now first of all you see outermost protecting layer is there. Now this particular protecting layer we will be calling as epidermis, and here I didn't draw the cells, but LME meristematic cells. Meristematic cells means has the division of labor more and more. Now the cells are divided repeatedly, so or divisionally will be active. So such kind of cells only we will be calling as meristematic cells. Epidermis means if you take this particular anther all cells are meristematic. On the side here is the conjunctive tissue. So here filament will be there. Now the entire one is young anther, and definitely the filament. Filament and anther both are attached at the conjunctive tissue only, no. So we have connective tissue. Okay, so this particular connective tissue, one important bit what they have given, no, connective, within the connective tissue what we can observe, no. So this particular connective tissue contains which one, vascular bundles. I have seen, so this particular bit in some other places, this particular connective tissue contains vascular tissues. If you don't forget, means here how here these vascular bundles are present, even myself also don't know, but author saying that this particular connective tissue, connective tissue means a filament that connects to the anther, no, there also they observe the presence of vascular bundles or the vascular tissues. Vascular tissues are nothing but xylem and phloem only, no. Now here later meristematic cells are there. At each corner, no, so cells become large. And what are the cells or become large and very active, no? These particular cells only our subject point of view we are calling as archisporial cells. These archisporial cells divides how? Periclinally. Already information we see, no? Periclinally divides. Periclinally division so definitely two cells of formation now. One by one. Now, two cells one by one form. Next one, when this particular archisporial cells or archisporial cell divides periclinally, up two cells of formation takes place. These two cells names are primary parietal cells and primary sporogenous cells. You see there. Totally 3 cells are there now. 3 cells cells together are there now. Primary parietal cells. After that, one and only the cell is now inside. Inside pattern. Inside cells, no, or inside a cell, not cells, cell. That particular cell we will be calling as a primary sporogenous tissue, or the tissue or cell. Author mentioned as primary sporogenous tissue. Tissue means group of cells, no, but here one and only the cell is there. At the time we cannot, so thereby primary sporogenous cell let us take. Primary sporogenous cell is there and primary parietal cells are there. Primary parietal cells when divides periclinally and anticlinally means definitely endothelium, middle layers and tapetum, definitely condipa formation takes place. Next one, when this primary sporogenous tissue divides by mitotically, so at the time now we can see the development of sporogenous tissue or microspore mother cell tissue. So that particular mother cell or the primary microspore mother cell or porum mother cell divides by mitotically and leads to the formation of tetrads. Already we have seen, no? Now all these tetrads are covered by means of callose layer. So this particular callose layer degenerated by the callase enzyme which is secreted by the tapetum. So already this entire information, so we have seen. Here, right. Now let us start to discuss the structures of pollen grains tetrads of angiosperms. So here, first of all, let us concentrate on this one. Here, பார்க்கும்போது இங்கே 3 cells, 3 pollen grains மட்டும் தான் இருக்கு, but one more அந்த side இருக்கு. போர்டுக்கு அந்த side. Okay, ma. So that's why here 3 only will be visible very clearly. So this kind of shape of this pollen grains, no, tetrahedral condition. So very familiar in a dicotyledons, means this kind of tetrahedral shape of the pollen grains we will observe. Pollen grain tetrad means 4 pollen grains, suppose if present together we will be taking as tetrad. Now you put tetrahedral shape of pollen grains or the tetrad we can see in mostly dicots. Next one, isobilateral. LMA seems to be same and this kind of arrangement. So 4 will be appeared and that kind of arrangement of tetrads we will be taking as isobilateral. So familiarly we can observe in monocotyledons. Next one, 3 mattum appear, but here both 2 cells are beside to the beside and one will be below the 2 cells. So decussate type. Next one, T-shaped. So here one line and here 2 cells. So thereby T-shaped tetrad arrangement, and one more last and final one is linear எல்லாமே ஒரே லைன் மாதிரி தான் இருக்குதுன்னா linear tetrad arrangement. So but here what are the main important points, or what are the main important points we have to discuss under this one? No, now you see there all the above types of tetrads are found in Aristolochia elegans. Aristolochia genus and elegans species name. Aristolochia elegans. In Aristolochia elegans, அங்கே இப்போ 5 types we are observing now. எல்லாமே இருக்கு சொல்லியாச்சு, but how it will be all types we don't know. But so all types of tetrads are found in Aristolochia elegans. Linear type we can see, tetrahedral we can see, isobilateral we can see, and desiccate, decussate and T-shaped. Likewise, there are different types of tetrads arrangement of the types we can see in Aristolochia elegans. But the most common type of tetrad is tetrahedral. So means in a monocotyledon, in a dicotyledons, what is a type we are observing now? So such particular type only is most common type. Most common type is tetrahedral arrangement of pollen grains. Next one, some facts. So some facts of the pollen grains. So here one fact only here I brought for you, so but still so many are there, approximately 9 are there. In the next class definitely I will give you. So what are the facts means? So first of all, first fact, some facts about the pollen grains. First of all, first one is translator apparatus. Which one? Translator apparatus in Asclepiadaceae. Simply Asclepi or Asclepiadaceae. This is one family name. Family name. Best example, Calotropis. Calotropis means one plant, if you pinch the plant, that milk that particular milk will not fall, should not fall in ice. Ice will go off. Likewise, parents will say no. Adi. Did you remember this one? Calotropis. Inge Calotropis nalli, and one more Archidaceae family members. There what we can observe, pollen grains are joined together to form a bag like pollenium. So means here pollen grains, what are the pollen grains are liberated or formed in the like Asclepiadaceae means Calotropis family and Archidaceae family members. No, all pollen grains are joined together to form a bag. இப்போ பேக் லைக் ஸ்ட்ரக்சர் ஃபார்மேஷன் மீன்ஸ் ஹியர் எல்லாமே ஒரு பேக் நிலை போட்டு பேக் உள்ள எல்லாமே போலன் கிரெயின்ஸ் இருக்குன்னா எப்படி இருக்கு ஸ்ட்ரக்சர்? ஸோ சச் கைண்ட் ஆஃப் ஸ்ட்ரக்சர் வி வில் பி டேக்கிங் ஆஸ் பொலினியம். Polonium. Just what is the point here you have to remember, no? In Asclepiadaceae or simple one, Calotropis and in Archidaceae members, no, pollen grains are joined together and form a bag-like structure is known as polonium. This particular polonium of Calotropis is a constituent of translator apparatus. So that particular when the particular all group of pollen grains present together and form a bag, you know, that particular one we can see in a calotropis. So Anthar he was he was considered that one as translator apparatus. This is one of the facts in pollen grains. So remaining in next videos I will give you. So try to watch this entire video. Easy da, easy da, but here I have approximately I have taken half an hour time. But try to learn now. So what is happening in the first cell and later what are the divisions, later what are the divisions, each and every point, you know, if you write notes, so thereby very exclusively you can save in your mind, so thereby no one will confuse you. So thank you and be safe at home.